All right, good morning, ladies and gentlemen. Hopefully you have had a chance to talk over with your group members how to actually do this lab. So we're going to be taking a look at photosynthesis and carbon dioxide use. So here I have some distilled water, and I'm going to add into it a chemical indicator that is called bromethymol blue. Bromethymol blue is blue in a neutral pH or in a basic pH. So this can be used as a pH indicator. And it turns yellow in an acid. Remember that you are blowing out carbon dioxide, and this equation comes up so many times in biology class. This is what ocean acidification is, because we're releasing too much carbon dioxide into the air and it's mixing into our oceans, and our oceans are becoming more acidic. It's why you have to exhale so that your blood pH doesn't drop. You don't want all that carbon dioxide that the mitochondria are producing to collect in your blood and lower the pH. That damages your proteins. Think about protein folding and tertiary structure. Okay, so what we're going to do today, I'm going to blow some bubbles. Hopefully this won't take long. If it takes a long time, go ahead and speed up the video right now. Oh, and you cannot see because I'm wearing, give me a second. I need something white. I'm looking for something white. Here we go. I'm grabbing something white over here, everybody, so that you can see. It didn't even occur to me that I was wearing something dark. I'm going to wear a bib today. Look at that. I'm looking good. You're like, oh my gosh, she's insane. Okay, so you can see the blue. It's going to take me a moment. Feel free to speed up. Oh, it's not taking that long. Can you see the color changing? We're already seeing it at the neutral color where it's going between blue and yellow, so we get green. I hope you're speeding this up because this will be hilarious fast. All right, do I have a yellow enough color? I think so. Can you see that that's a nice yellow color? I'm going to go ahead and pour that into my test tubes. So hopefully you can see me. Maybe I should do this. I'm going to scooch this really close and do it as much in front of the white background as possible. So I'm going to go ahead and pour the yellow water in. And I want to make sure that I can tell that that's yellow. I might need to add a little bit more of the bromethymol blue to it, just to make sure that we can really tell the color. Can we see that that is definitely not blue, at least? And then I'm going to go ahead and add, here are my underwater plant. This is Elodea. So I'm going to add a sprig of Elodea. Here's my first sprig. I'm going to go ahead and push that down. And then we're going to put a stopper. You'll just have to trust me that I'm going to put a stopper on. I don't have the stopper handy. But you can see that it's still that yellow color. And then I'm going to do that 3 times in 3 different test tubes so that we can watch it progress over the day today. I'll add a second one here, shove it down. And then I'm going to place these in the light. Woo, don't want to spill on myself. I guess I could have taken off my bib now. There, no more need for a bib. All right, and then one more. We're going to fill with yellow water. I'm just repeating the experiment so you can be sure that this is legitimate. It's like adding extra trials to the experiment. I'm adding more color so we can see it a little bit better. So hopefully you can tell that's definitely not blue. It's more at the intermediate level. And remember, there's lots of carbon dioxide in the test tube right now. And so what I'm doing is placing the plant in there. We're going to place the plant in some sunlight. And then we're going to watch it to see if it photosynthesizes. In addition, I don't know if you guys can see this. I asked the aquarium store for some snails. And what I'm using is water, do you see the blue color? This is water that came in the bag with the plants. That water came in the bag with the plants, and that's good water for these snails. If I put these snails in distilled water, it would be hypotonic and they would die. So I put them in water that had the plants in it, but now I'm going to put them in the dark so that there's no photosynthesis or anything going to be happening in the light. So I'm going to go ahead and put them in a cabinet in the dark over here. We'll leave them here for several hours. And then we'll come back and we'll test what color that water ends up, whether it's blue or green. Now I'm going to walk you over. I'm going to put some, you'll just have to trust me that I'm putting some corks on these so that none of our gas escapes. And I'm going to go ahead and set them here in the window. And then we'll check on those periodically over the next few hours. All right, I'm going to go ahead and pause here. I've got to get a test started. Okay, guys, I had to move our plants because I don't have power in my lab at the moment. I'm not sure why I don't have power. So I moved the plants and I added some stoppers to them so that none of the gas would escape. So the plants now are right there. You can see the high school parking lot. And I added a lamp because it's a cloudy day outside. And so I don't know if they're going to get enough light for us to be able to see them change color well. But we'll let them go and I'll keep coming back. I can already see bubbles rising in them. That means that oxygen is being produced. We'll come back a few hours later.